So, there are around 30,000 recipes in your genome. And during every cell division, all those recipes are copied. So you understand already that sometimes something might go wrong. Let's have a look. Imagine that in this recipe for hemoglobin that we saw in the previous video lecture, something happens. This A of the codon GCA changes into a C. We call this a mutation. So the codes now read GCC instead of GCA. What amino acid is this coding for? What will be the effect of this mutation? Let's have a look at the genetic code. Nothing. It still codes for alanine, right? There's no change in the protein. Whether there is a C or a G at this position, the same amino acid is incorporated. There's no effect of this mutation. There is no way for natural selection to evaluate this mutation. Now that actually is an important conclusion. There are many mutations that have no effect at all. Okay, let's have a look at another position. Imagine now that this G changes into a C. So instead of GCA, the code now reads CCA. What amino acid do we have to incorporate? Going to have a look at the genetic code. Proline, right? This C at this position leads to a different amino acid. And do you think that is good? Probably not, right? It's a bit like me changing a car. If I detach somewhere in the engine a tube and attach it somewhere else, I probably broke the car. This hemoglobin was working fine. A change will probably only make it worse. And indeed, that's true. We saw already that many mutations do not have an effect at all, but if they have an effect, they're usually bad. But occasionally a mutation happens that makes the protein better. And I, of course, chose an example in which this is the case. This hemoglobin is not a human hemoglobin. It is hemoglobin from a bird, from the grey-lag goose. And this goose is found all over Eurasia at low altitudes. And this hemoglobin, with the C at this position, that is the bar-headed goose hemoglobin. And this goose is found in Central Asia and migrates every year over the Himalayas to overwinter in South Asia. And what does hemoglobin do? It binds to oxygen in the blood. That blue blob in the middle clearly is an oxygen molecule. And this bar-headed goose hemoglobin with the proline that binds much better to oxygen than that of the grey-lag goose. Have a look. In the 70s, scientists have shown that it takes much less pressure to saturate 50% of the hemoglobin in the bar-headed goose, look at the circles, the lower line, than it takes to saturate the hemoglobin of the grey-lag goose. Look at the triangles, the middle line. Now why would that be an advantage to the bar-headed goose? Of course, they fly at amazingly high altitudes with much lower air pressure and also much less oxygen. This goose can use a better hemoglobin. So, do you think that this mutation never happened in the grey-lag goose? Why don't all those grey-lag geese have a better hemoglobin? Yes, it doesn't need it at low altitude, right? Or to be more precise, it didn't cause more offspring. A goose with the old hemoglobin just got the same number of offspring. The mutation might well have happened, but it was not propagated by natural selection. It didn't matter for the grey-lag goose. Now, for the bar-headed goose, in contrast, such a hemoglobin would be an enormous advantage. While its colleagues were arriving exhausted at the other side of the Himalaya and had to take a rest, the bar-headed goose with this new hemoglobin could directly start building a nest and producing offspring. And so the mutation spread. Now, this is how evolution works. Mutations cause new variation, and if it is advantageous, it will be selected, and if not, it will disappear. And particularly at the end of the 70s and in the early 80s, scientists really had the impression that they understood evolution. We understand natural selection, we understand where new variation comes from, we understand evolution.